Okay, everyone on the line. So the first session, I will moderate the session with Tatsuya Morimoto from the Japan. So I'm from the China, the Third Military Medical University. Welcome to the session, recent advance in the cardiac myocyte regeneration. So the first let's go to the first speaker is Roger Fu from Simple. So the title is ASNS a mark for cardiomyocyte uh, de-differentiation and a gateway to regeneration. Dr. Fu, please.
Thank you. Thank you, Dr. Cheng. Thank you, Dr. Morimoto. Allow me to share my screen. Thank you. Good morning, everybody. The Fu Lab has, for many years now, been very interested in cell states and gene expression and gene programs. And in recent years, there are two publications that we have uh, been able to put together fortuitously looking at how cardiomyocytes roll down the hill and also up the hill. The title of the presentation given to the organization was the story about us of the marker going up the hill, published by Ike. But actually, it turns out that I would like to speak about the marker that is going down the hill, and I will explain why. In recent years, we have in Singapore, in NUS, been building a stem cell bank, and this is comprising of more than 1,000 and actually now 2,000 donors, where we are making iPS cells and performing deep clinical phenotyping on the cohort using genetically encoded sensors and also genomic safe harbors, and using a huge uh, expertise now that we have experience now that we have compiled with genomic computational tools. Studying iPS cells in cell village, carrying out DNA GWAS in a dish, integrating genomics towards target prioritization, and also then validating our targets both in vitro and also in vivo, and very much using CRISPR technology. For example, in our collaboration with Professor Corey Levine at the Washington University in St. Louis, Mick, who is the main driver of this project with our stem cell bank, has been looking at the targets of variants sitting inside distal enhancers interacting with heart failure genes. The reason why this project I thought was really beautiful was Mick took the approach where he carried out genome-wide CRISPR screening using cardiomyocyte differentiation, so rolling down the hill. Cardiomyocytes differentiation is a protocol that is well known and well established in the community. There are very specific markers for the successful differentiation, starting from OCT4 and SOX2 in the pluripotent stage to the markers that you see here, Mercury, UMS, to MSP1 as mesoderm stage, to progenitor stage with MYH6 and NKX2.5. So using this established protocol, carried out cardiomyocyte differentiation starting from pluripotent stem cells and deployed a human Brunello library that was a CRISPR genome-wide screen comprising of 77,411 single guide RNAs in a pool, where there are 4 single guide RNA targeting each protein coding gene. We made use of a cell model that had GFP that is driven by MYH6 promoter, so as to recognize green cells that are successfully differentiated to cardiomyocytes. And in this screen, when Mick was analyzing the data, there were very good positive controls that appeared in the screen relating to genes that were in very well-known pathways for cardiomyocyte differentiation. As negative control, neuronal markers were in the negative candidates of the screen. And there was one gene that stood out that had not been previously studied before for cardiomyocyte, which is NF2. NF2 is the gene which, when mutated, gives patients a condition called neurofibromatosis type 2. Neurofibromatosis produces hearing loss and balance problems, specifically giving rise to many tumors along nerves. And it's a very neuronal condition. Interestingly, we looked at the literature and found that there was no reported cardiac involvement in neurofibromatosis. So we asked ourselves the question why, as to our cardiomyocyte screen should produce the target NF2. I think we have the reason, and I'm happy to discuss that if anybody asks this question later. So in order to validate the candidate of NF2, we looked at the gene NF2 and found that there is a set of interesting domains in the protein of NF2. The firm domain is a domain which we will come up to mention later on in the slides, related to proteins that are very frequently found on the cell adherence junction. And what Mick did was to target using two sets of guide RNAs to create single clones of NF2 knockout. So in this case, clone 1 was an NF2 knockout where there was one single base insertion creating a frame shift. And in a second clone, he created a heterozygote or hemizygote knockout where in one clone, one allele, there was missing stretch of bases, and in another allele, one base pair missing. So he created two knockouts of NF2. And in differentiation of the wild type and knockout, he was able to show that the formation of mesoendoderm was largely and mainly disrupted in NF2 knockout cells, whereas the differentiation to ectoderm was unchanged. And he used markers to show also that in NF2 knockout cells, brachioendoderm markers were absent for cells that were being differentiated and rolling down the hill. This led to confirm that NF2 deficiency created the loss of mesoendoderm. Furthermore, in differentiation, not just towards mesoendoderm, but also to cardiac progenitor, there was a loss of HEN1 marker in day 5, and the loss of NKX2.5 as a marker of cardiomyocyte. And as mentioned, our cells that are green, indicating MYH6 expression or cardiomyocyte formation, this was absent in both clones of NF2 knockout. Confirming here that in NF2 deficiency, there was a loss of cardiomyocyte formation. So Mick went further to investigate what relationship NF2 might have to cardiomyocyte differentiation processes that are known. And loosely in the literature, there was some suggested evidence of a relationship between NF2 and YAP pathway. And as we know, YAP is very tightly regulated during cardiomyocyte differentiation. And Mick proved indeed that in the absence of NF2, so in both of the knockouts, phosphorylation of YAP was absent in mesoendoderm formation in early cardiomyocyte differentiation. But interestingly, Mick went to investigate further as to whether the canonical kinases involved in pathway of YAP signaling was changed, and he found an absence of changes in both phosphorylation of upstream kinases LATs as well as MST1. So we concluded at this stage that YAP seems to be altered as a result of knockout of NF2, but it does not seem to be involving the canonical pathways of LATs or MST1. nor did it involve the abundance of YAP itself. This is the point where Mick then went to investigate whether it was the localization of YAP that was altered. And indeed, we found that whereas in wild-type cells, YAP localized to the cytosol in the transition to mesoendoderm, whereas in knockout cells, YAP stayed in the nucleus, as you can see here. So we decided to pay close attention to YAP nuclear relocalization as a result of NF2 deficiency. And at this stage, we also took very close look at the transcriptome of the cells that were differentiating. And Mick was able to show that the differentiation of cardiomyocytes in wild type with the presence of NF2 created a very clear trajectory marked here in blue, where there were progressive markers of cardiomyocyte formation, progenitors, and beyond. Whereas in the absence of NF2, there was a transition that was changing in direction, specifically the loss of all cardioprogenitors and cardiomesoderm markers, so altering the trajectory of cardiomyocyte cell state. In analysis of the transcriptomes of the NF2 knockout cells, he was able to produce a explanation where NF2 seems to be required for lifting the roadblocks of OX4 and SOX2 in the journey from pluripotency to mesoendoderm, and also lifting the roadblocks of CDX2 and MSX1 at the point when Wnt was turned off in order for journeying further down to cardiomyocytes. So the conclusion at this stage was that NF2 is necessary to remove the roadblocks en route to cardiomyocytes as the cells were journeying down the hill. Back to the molecular mechanisms, Mick was able to show that in the absence of NF2, there was a loss of interaction between NF2 and YAP because NF2 interaction with YAP in wild type was present in mesoendoderm stage and also validated with proximity ligation assay, where in the presence of NF2, there is close correlation with and close co-localization with YAP, whereas in the absence of NF2, YAP and beta-catenin were no longer co-localized, implying that this was the relationship between NF2 and YAP at the cell adherence junction marked by beta-catenin. So YAP NF2 complex is at the adherence junction in mesoendoderm during day 2. So at this stage, we went back to look at the NF2 protein, and I mentioned the firm domain, which is a domain that is found on many proteins that are situated in the cell adherence junction. Within the firm domain, there is something called the blue box domain, where this is a particular domain with very characteristic motifs of 7 amino acids. So we paid some attention to that. In addition, in the literature, there is also description of how NF2 can exist either as a closed protein or an open conformation, and both of which have significant interactions with other proteins relating to its tumor suppressor function, giving rise to neurofibromatosis and benign tumors. And this was also highly related to a phosphorylation in serine 518 position, which regulates the open and close formation of NF2 generally. So on the background of the knockout cells, undertook the or the re-expression or introduction rescue experiment with either wild-type NF2 with a flag tag or a series of mutants, including a mutant of the serine 518 to alanine mutation, so a non-phosphorylatable version, or a phosphomimetic, which is a S518D mutant. He also undertook the mutation to the firm domain in the blue box region by either deleting the 7 amino acids or mutating all 7 amino acids to alanine. And on this background, he was able to show that the wild-type rescue recapitulated the cardiomyocyte formation, as you can see here, with green cells that were formed with wild-type rescue. And interestingly, both the phosphomutant as well as the phosphomimetic were able to also rescue cardiomyocyte formation, but not the blue box domain mutants. So not the mutant where blue box domain was deleted, nor the blue box domain mutation to alanines. And he was also able to show the same result of the rescue of the YAP localization rescued by wild type as well as the phosphomimetic and the phosphomutant, but not the blue box domain mutants. And equally, he was able to show that the interactions between YAP and NF2 were changed as a result of rescued as a result of the wild type expression, phosphomutant expression, but not the blue box domain mutants. So we concluded the work by saying that the NF2 blue box domain is necessary for cardiomyocyte differentiation, and it seems also necessary for YAP localization into the cytosol during mesoendoderm transition. And it's also necessary for the YAP NF2 complex formation together with MOT, which I did not spend too much time describing here. YAP at the adherence junction seems unrelated to phospho NF2 at the serine 518 position, and it does not seem to be related to closed or open conformation of YAP. And we also proved at this point by reviewers' request the interaction between MO3 and YAP were also lost in the NF2 knockout, but present in NF2 wild-type cells. What we were not able to fully address was whether MOT and mutant NF2 were still interacting at the situation when the blue box domain was mutated. So in summary, we have found a pathway in which cardiomyocyte differentiation, starting from pluripotent cells, required NF2, which localized YAP from the nucleus into the cytosol, parked together at the cell adherence junction with NF2 and MOT, allowing mesoendoderm genes to be switched on for the transition towards mesoendoderm. In the absence of NF2, this transition of YAP is unable to take place. YAP remains in the nucleus and as a result blocks the expression and programs of mesoendoderm genes and pushing the cells towards a different form of cell differentiation. So at this point, the lab is excited because we are investigating whether there are options for YAP regulation, especially in the realm of cardiomyocyte regeneration, using the knowledge that we now have with NF2-based therapeutics, for example, targeting the blue box domain. Thank you very much for inviting me to speak. My lab is based in NUS in Singapore, and we have some affiliation to A-Star in the Biopolis, and we welcome you to visit us in Singapore. Thank you.
Thank you, Dr. Fu, for your wonderful speech. Dr. Fu found the role of the NF2 in the cardiac regeneration. So we go to the question. I have a short question. So how about the expression of the NF2 in the adult cardiomyocyte and the other cells in the heart?
Thank you, Chunyi. NF2 definitely is present in adult cardiomyocyte. It actually doesn't change in expression very much. It's a very stable protein we find parked at the cell adherence junction. So the main changing component in cardiomyocytes we find is definitely the YAP protein. So this is probably the reason why there's many studies targeting YAP for a lot of cardiomyocyte stress situation.
So how about the expression with the neonatal cardiomyocyte for the adult cardiomyocyte? Is
Yes,
higher or lower?
yeah, so we find the same also that NF2 doesn't seem to be altered very dramatically in expression. What we think probably is important physiologically or maybe developmentally is that when cardiomyocytes are formed and there is cell-cell interaction, and the cell-cell interaction at the cell adherence junction probably alters components of cell adherence and cell surface or under the cell surface interaction. So it alters, therefore, YAP localization in order to push cardiomyocyte differentiation.
Okay, thank you. So, Dr. Morimoto, do you have any question?
Yes. Yes. Uh, do you check the expression level and localization of your plan for the heart failure developments?
No, so we haven't done that, Taswia. So this is something that is also studied very widely in our community of researchers, such as Jim Martin. So they have seen differences in YAP localization or YAP abundance and YAP also phosphorylation. We didn't look at this ourselves.
Thank you very much.
Okay, thank you. Thanks for Dr. Fu's speech. Okay, next we go to the second speech from the Fuai Hospital, Dongcheng Cai. So the title is about the Fox K family drives the cardiomyocyte glycolysis and heart regeneration. Okay, Dr. Cai, please. Dr. Cai, no voice. I couldn't hear you.
Okay, can I see my
Okay,
screen?
okay, it's fine.
Okay, you cannot. Okay,
Okay, fine. It's fine.
okay.
Mhm.
Due to the time conflict of Professor Neil, so I'm here to give the presentation on his behalf. So my name is Dongcheng Cai, a postdoc from Professor Neil's lab, and thanks for the introduction by Dr. Zeng. And it's an honor to share our latest research about Foxy family in cardiomyocyte glycolysis and heart regeneration. So as we all know, as we all know that heart failure still remains the leading cause of death worldwide, and currently there are nearly 60 million heart failure patients globally, and the 5-year mortality is over 50%, which causes a huge burden. The heart failure is often caused by myocardial infarction, and this is due to the occlusion of coronary arteries, leading to ischemia and hypoxia of the myocardium. As a result, large amounts of cardiomyocytes were lost in a short period of time, and the lost myocardium cannot was replaced by the fibrotic scar, and eventually this lead to cardiac dysfunction and the development of heart failure. The underlying reason is limited capacity of proliferation for adult cardiomyocytes, and they cannot replenish the lost myocardium in a timely and effective manner. But for the neonatal hearts, such as the neonatal mice, they still maintain the regenerative capacity, while endogenous cardiomyocyte proliferation in response to injury, such as a myocardial infarction. So, if we can induce endogenous cardiomyocyte proliferation and provide enough functional cardiomyocytes, and we can reverse heart failure and improve the cardiac function. In fact, studies in the past decades have proven the feasibility of this idea. So when we talk about heart regeneration, we should think about who involves. So if we consider cardiomyocytes as the seeds for in this process, then the surrounding microenvironment, including the extracellular matrix and the inflammatory stimulus, may act as a soil that can support the proliferation. Actually, the past few years, our lab have published some work in this area. So the research goals in our lab is to decode the regulatory mechanisms of cardiomyocyte proliferation and identify the potential therapeutic targets for heart regeneration. As we just mentioned, the proliferative capacity of cardiomyocytes decreases with development. So this is caused by many factors. One of them is the metabolic shift. After birth, the metabolic substrates of cardiomyocytes shift from glucose to fatty acids, but this process also generates increased mitochondrial reactive oxygen species, which can cause the nuclear DNA damage and resulting in cardiomyocyte cell cycle arrest. So if we could change the energy metabolism pattern of cardiomyocytes, it might be possible to promote the cardiomyocyte proliferation. So this is where the FOXQ family comes in. So the FOXQ family comprise two members called FOXQ1 and FOXQ2. About 6 years ago, that labs have found that FOXQ1 and FOXQ2 can enhance aerobic glycolysis by inducing the expression of glycolytic enzymes. So the idea comes to us that can FOXK family regulate cardiomyocyte metabolism and proliferation? So we first isolated the primary most cardiomyocytes and overexpressed the FOXK1 and FOXK2, and we detect the metabolic pattern of cardiomyocytes with Seahorse analysis. As you can see here, that overexpression of FOXQ1 and FOXQ2 can significantly enhance the glycolytic capacity, as revealed by the increased rate of extracellular acidation. And we also performed the metabolic flex analysis with isotope labeled glucose, and we can the mass spec. And here you can see that it has overexpressing FOXQ1 and FOXQ2 levels of glycolytic metabolites were significantly increased. So the next question we want to address is whether FOXQ1 and FOXQ2 can promote cardiomyocyte proliferation. So in the cultured cardiomyocytes, we found that the overexpression of FOXQ1 and FOXQ2 can increase the proportion of proliferating cardiomyocytes. Here we use the phosphorylated histone 3 and Cas87, kinase P as kinase B as proliferative markers. And conversely, if we knock these two factors down in cardiomyocytes, the capacity of proliferation of cardiomyocytes was significantly decreased. So next we test the connection between FOXK regulated cardiomyocyte metabolism and proliferation. Here we used 2-DG is a deoxy-D-glucose is a glucose analog can inhibit the glucose to treat the cardiomyocytes. And we found that the treatment with the 2-DG can significantly inhibit cardiomyocyte proliferation induced by FOXK1 and FOXK2. This indicated that FOXK1 and FOXK2 induced the metabolic shift is important for cardiomyocyte proliferation. So here we can make a brief summary, and FOXQ1 and FOXQ2 can promote cardiomyocyte proliferation and glycolysis. And next, we establish the inducible cardiomyocyte-specific FOXK1 and FOXK2 knockout mouse model. On the postnatal day 1, we induced the myocardial infarction via LAD ligation. On 28 days after MI, here you can see that the ligaments can regenerate completely. However, in FOXQ1 or FOXQ2 condition knockout mice, the fibrous scar was significantly increased. We also validated the cardiac function, and here you can see the cardiac function, the knockout mice is significantly lower than the litmates. 5 days after the infection, we detected the cardiomyocyte proliferation rate, and we found the FOXQ1 or FOXQ2 condition knockout in cardiomyocytes, the cardiomyocyte proliferation was significantly diminished. So we can make the second summary that in vivo we found that FOXQ1 and FOXQ2 are required for nucleotide regeneration after myocardial infection. So the next question is we want to ask is what's the mechanism? How these two transcription factors regulate cardiomyocyte glycolysis and proliferation. So here we use a catalytic analysis to analyze the binding sites of FOXQ1 and FOXQ2, and we found that these two factors can both bind to the promoter region of hypoxia-inducible factor, which can control the cell proliferation and metabolism. So next, we knock down this factor in overexpressed cardiomyocytes, and we found that we measured the cardiomyocyte glycolytic capacity and the proliferation. Obviously, knockdown of HIF-1 alpha blunts these two processes, indicating that HIF-1 alpha sieves are crucial for FOXQ1 and FOXQ2 induced cardiomyocyte proliferation. So next, in our catalytic analysis, interestingly, we found that FOXQ1 and FOXQ2 can may also drive cardiomyocyte proliferation by binding to the promoter region of cell cycle related genes. Here is a gene ontology analysis of the binding sites of FOXQ1 and FOXQ2 in cardiomyocytes. So next, we did a joint analysis of catalytic and RNA-seq, and we found that CCMB1 and CDK1 may serve as downstream factors. And we tested this idea in primary cardiomyocytes and showing that FOXQ1 and FOXQ2 expression can significantly induce the expression of CCMB1 and CDK1. And then we knocked down CCMB1 and CDK1 in FOXQ1 and FOXQ2 expressing cardiomyocytes, and this results a significant inhibition of cardiomyocyte proliferation induced by FOXQ1 and FOXQ2. So here we think that FOXQ1 and FOXQ2 can promote cardiomyocyte proliferation through dual mechanism by regulating cardiomyocyte metabolism and drive the progression of the cell cycle. So finally, we want to know whether FOXQ1 and FOXQ2 can promote heart regeneration in adult mice. So to drive this, we induced myocardial infection in adult mice and administered the adeno-associated virus to overexpress FOXQ1 and FOXQ2 specifically in cardiomyocytes. And the results show that after the surgery, the cardiac function diminished. And however, for the group in FOXQ1 and FOXQ2 expressing hearts, the cardiac function continued to recover in the following weeks. And next, we did the trichome staining. The results show that overexpression of FOXQ1 and FOXQ2 can significantly blunt the fibrotic scar formation after myocardial infection. More importantly, we found that overexpression in cardiomyocytes can significantly enhance adult cardiomyocyte proliferation after myocardial infection. So at the last, I want to use this cartoon to make a final summary that we found FOXQ1 and FOXQ2 can promote cardiomyocyte proliferation and accelerate the cardiac function recovery after myocardial infection. by promoting cardiomyocyte cell cycle progression and the metabolic shift. So finally, the project was completed and under the guidance of Professor Nie, and we also thank the funding report. So I will stop here. And thanks for listening, and welcome to our lab. Here is our lab's website. Thank you.
Okay, thanks, Dr. Tsai. Our interesting result. So I also have a questions about the, you have the great study for the interaction between the endothelial cells and the cardiomyocyte. So this one you find in the cardiomyocyte, you find the FOX1, K1, and the FOX K2. Did you check the interaction between this gene on the endothelial cell?
Thanks for your question. You say
Yes,
that's the expression level?
I mean, for the artery or the muscles, there's some effect on the cardiomyocyte regeneration. So for this gene, the genes, you just modulate this gene in the cardiomyocyte. So how about is there any effect on the endothelial cells?
Actually, we did some experiments in the cell cells. We expressed them in the cell cells, but we don't have the increase of in the cell proliferation. But if we knock them down in the cell cells, the proliferation was decreased. So and the expression distribution in the heart, I think FOXK1 and FOXK2 is ubiquitously expressed, so it's a relative high level. But in the development with the development in cardiomyocytes, the FOXK1 and FOXK2 were decreased with development.
Okay. Any
Thank you.
questions? Dr. Morimoto, yeah, you have any question?
Thank you very much for interesting presentation. So does FOXK expression level changes in the human heart failure model?
We didn't check the expression in human sample, but we checked them in the in mouse hearts after myocardial infarction. That we found that FOXK1 and FOXK2 were downregulated after myocardial infarction. So we did use the strategy to overexpress them in the adult hearts. Thank you.
Thank you
Okay,
very
Dr.
much.
Tsai, thank you. So I transfer the moderator to Dr. Morimoto. Dr. Morimoto, you please.
Okay, thank you very much. Let's move on the next speakers. Next speaker is Yang Hori from Korea. His title is Emerging Mechanism of SL2 Inhibitors Effect on Cardiometabolic and Renal Disease. So please start your presentation.
Thank you, Chairman. Can you hear me?
Yes.
Thank you. It's a great honor for me to have this presentation in this wonderful symposium, and I'd like to thank the organizer for giving me a chance. And as this session is mainly focusing on the basic research, whereas my talk includes some clinical aspects as well. I'm not sure if it's fully aligned with this theme of this session, but I hope this kind of idea can provide some useful insights for the basic researchers. As you know, SGLT2 is a transporter in the kidney proximal tubules, as you can see here, and increased SGLT expression is found in diabetes patients. So usually diabetes patients has higher glucose in the kidney tubules, so it has to absorb from the SGLT2 transporters like here. So the concept of SGLT2 inhibitor, as shown here, to block the glucose reabsorption from the kidney, so it can induce glucoseuria. So about 300 kilocalories per day of glucose is going out from the kidney through urine, so it can decrease glucose levels as well as body weight reduction and blood pressure reduction as well. Recently, many clinical studies found the protection of cardiovascular diseases and diseases from the diabetes patients and also non-diabetic patients. So SGLT2 inhibitor is recommended as a first-line treatment for CKD or cardiovascular disease, especially in heart failure as well. So the left side is the diabetes guideline and the right side is the cardiovascular heart failure guidelines. So SGLT2 inhibitor is considered as the first line. And also from last year, KDIGO guideline, which recommends the CKD drug treatment, SGLT2 inhibitor is considered as first-line drug therapy. But the mechanism is like it can block glucose reabsorption and also sodium reabsorption. So SGLT2 inhibitor can induce osmotic diuresis and natriuresis. So many kind of cardiovascular protection and kidney protection can be explained by these two mechanisms. Using SGLT2 inhibitor, so sodium goes to the distal tubule like a low concentration, so it can modulate the tubular glomerular feedback mechanism, so it can change the glomerular pressures shown here. So maybe this can lead to glomerular structures, and also it can induce natriuresis with diuretic mechanism, so it can decrease the blood pressure as well. So because of the glucosuria, it can reduce the glucose levels as well. So usually when we use glucose SGLT2 inhibitors in patients with diabetes or non-diabetes, these two things are the direct mechanism of this drug. But because of these effects, it can induce uricosuric as well. And also because every day 300 calories are going through our body to urine, so negative calorie balance can modulate our metabolism in the body, and also other things happens. So many kinds of mechanisms are proposed by many researchers using animal models and cell models as well. So first, using SGLT2 inhibitor can induce hematocrit. The large clinical trials, after mediation analysis, found that most of cardiovascular protective effects driven by the increase of hematocrit or hemoglobin shown here, like 50% of these cardiovascular protective effects are mediated by these two factors. And also recent AMPA Heart CardioLink trial showed that erythropoietin levels are significantly increased after using empagliflozin or SGLT2 inhibitors. And why these things happen? Many animal data and cell data found that usually SGLT2 emission can occur in the proximal tubule, usually cortex of the kidney. And because of these things happen in the medullary region of the kidney, because one expression in the kidney tubules can lead to the local hypoxia in the medullary region, which induce the HIF2 alpha, and that can lead to the increase of erythropoietin levels. And because of the increase of erythropoietin can make many kinds of beneficial effects, including the inducing erythrogenesis and utilizing iron and oxygen delivery can improve in the kidney or heart. And also there's a research that it can induce circulatory progenitor cells in the human blood samples. Another thing after using SGLT2 inhibitor is the increase of ketone bodies. As you can see in these two graphs shown here, after using acute one single dose of SGLT2 inhibitor or the chronic using for 1 month can both increase the beta-hydroxybutyrate ketone body levels in the humans, regardless of the diabetes status. And because of this increase of ketone body may be explained by the decreased level of insulin, because insulin is a potent suppressor of ketogenesis. And there are three kinds of ketone bodies, and most of the soluble ketone body called beta-hydroxybutyrate is a useful source for energy from the mitochondria. And also, there are many research showing that not only energy source, but act as a single molecule by the ketone bodies. It can activate HCAR2, which is a G protein coupled receptor, and that can lead HDAC inhibition as well. There's some paper that high ketone levels are a protective role in the metabolic diseases. Our group conducted this kind of research using the with protective cord shown here. People who have spontaneous ketonuria in their health checkup data from the urine, that means ketone body levels are increased in those patients. And those people with high ketone body levels, they have protective effects against the development of diabetes shown here. And comparing to the non-ketonuria versus ketonuria patients have lower postprandial glucose levels shown here, indicating that they can utilize glucose very well. And sorry for the complicated figure, but what I want to stress in this picture is that our group also confirmed that ketone body can be produced not only from the liver, but also from the intestine and also kidney after using SGLT2 inhibitors that can reabsorb ketone bodies from the urine. So that can lead to increase of ketone bodies in the whole body. And also, from the circulation research, when they infuse ketone body into the chronic heart failure patients, that can directly improve the cardiac output, which is the cardiac function in patients. As you can see, this is a crossover study design when they infuse ketone bodies for a couple of hours, and then they measured cardiac output shown here that can dramatically increase the cardiac output from the chronic heart failure patients. Also, there is a research that ketone can inhibit interleukin-1 beta from the Nature Medicine paper shown here, and that indicates that maybe ketone body can act as a inflammasome inhibitor. And inflammasome is a very important target for cardiovascular disease, showing in the right panel. From the CANTOS trial, they used monoclonal antibody targeting I1 beta. And when they used this blocking I1 beta antibody for cardiovascular disease patients, their cardiovascular events were decreased by 14%. So our group conducted a prospective clinical trial with using SGLT2 inhibitors, and then we measured macrophage inflammasome activities from the human blood samples. As you can see, comparing to the control group, SGLT2 inhibitor can significantly inhibit inflammasome activities from the PBMC samples. And we confirmed that this kind of inhibition of inflammasome can be driven by the lowering glucose levels and also uric acid and insulin levels as well. So, and also increased by the beta-hydroxybutyrate by the SGLT2 inhibitor that can inhibit inflammasome activities from the human macrophage samples. And also there's a research that ketone can directly protect kidney disease from this cell metabolism paper shown here. And because in diabetes patients' kidney samples, they have mTOR hyperactivation were found, and this kind of marker is the important pathogenesis for the diabetes kidney disease. And they use HMCS2 knockout mice, which is a very important enzyme for production of ketone bodies. And when they look at these knockout effects by using SGLT2 inhibitors, SGLT2 inhibitor can protect kidney from these knockout mice model, but when they treat SGLT2 inhibitor in the HMGCS2 knockout mice, their protective effects by the SGLT2 inhibitor were abrogated, which indicates the ketone body production is really important. And also, comparing to the normal patients, type 2 diabetes patients have decreased glucose uptake by the PET-CT imaging shown here, indicating that glucose utilization is impaired in type 2 diabetes. But when we use SGLT2 inhibitor in DBD mice model, that can increase the glucose and also it can improve the ketone body oxidation as well, indicating that SGLT2 inhibitor can improve cardiac energetics. And also, regarding with the previous can improve cardiac reverse remodeling from the cardiac MRI studies as well. And maybe it's not confirmed yet, but maybe this kind of direct cardiac inflammation and fibrosis effects are driven by the impact on SGLT2 inhibitor in sodium hydrogen exchanger inhibition, but it's not confirmed yet. Recently, many papers showing that SGLT2 inhibitor can have potential anti-arrhythmic effects shown here. It can reduce the arrhythmia in very high-risk patients. And you can see the SGLT2 inhibitor can drop more than 30% of incidence of arrhythmia. And maybe a recent paper showing that SGLT2 inhibitor can reduce the sympathetic overactivity from the mice model shown here. It can reduce afferent renal sympathetic nerve system activation, and also it can lead to the reduce the renal expression of SGLT2 inhibitor expressions as well. And recently, metabolic reprogramming or starvation mimicry effects by the SGLT2 inhibitors were proposed by many review articles, including Nature reviews and Circulation as well. And this concept also confirmed with this kind of human tissue findings shown here. When you look at these Western blot from the kidney tissues, you can see compared to the DBDB control mice, which is mice model, when they use this mice model, an expression of SGLT2 inhibitor is increased, and also sirtuin levels are decreased. But when you use the SGLT2 inhibitors in the DBDB mice model, SGLT2 expression is not that changing, but SIRT1 is improved, indicating that SGLT2 expression and SIRT1 is kind of correlated. And also, when you look at the human tissues of kidney model showing here, SGLT2 expression and SIRT1 expression are inversely correlated. And this indicates that SGLT2 transporter is an energy surplus sensor linked with sirtuins such as fast-liking signal molecules. And also, our group confirmed that using the kidney liver tissues, when glucose enters to the glucose transporter such as GLU4 or SGLT2 transporter, it can convert into UDP GluNAc through pentose phosphate pathways, and it can induce oligomethylation of various cellular proteins. As you can see, when we induce SGLT2 expression in the cell line models, you can induce oligometh protein as well. And also in the liver of human liver samples, we also found correlation between SGLT2 expression and oligometh expression are highly correlated. So this kind of energy source surface condition can mimic by the SGLT2 expression. So SGLT2 inhibitor using can mimic calorie restriction through various kind of mechanism, including autophagy induction. So because it can induce glucosuria, it can mimic system, and that can induce many kinds of calorie restriction related enzymes such as AMP kinase, SIRT1, and mTOR inhibition. So we also confirmed after treating SGLT2 inhibitor, it can induce AMP kinase and induce autophagy through TFAB mediated pathways. So many research review articles showing that nutrient deprivation signaling, including or certain families, can be activated by the SGLT2 inhibitor, but also mTOR inhibition can achieve by this using these drugs. So recent animal data showing that SGLT2 inhibitor can reduce myocardial infarction area, but when they inhibit AMP kinase or inhibit autophagy by using chloroquine or compound C, you can see the protective effects by the SGLT2 inhibitor are abrogated, indicating that this kind of autophagy-related mechanisms are among dependent on the SGLT2 therapeutic effects. And recent large-scale proteomics study from the human trial after using SGLT2 inhibitor also confirmed this kind of things. Many proteins induced by the use of SGLT2 inhibitor are linked to autophagy induction and SIRT1. And also recent empagliflozin large EMPEROR program, their proteomic analysis also confirmed that many proteins increased after SGLT2 inhibitor, like EPO or IHBP1, those are regulated by the SIRT1 as well. And also recently, cellular senescence is an important mechanism for the cardiovascular and metabolic disease as well. And when you see these cartoons, the blue cell means senescent cells, and senescent cells accumulation can induce multi-organ dysfunction, including cardiac and adipose tissue and kidney as well. And recent paper from Nature Aging from the Japanese group also found that anti-aging effects of SGLT2 inhibitor can be confirmed using mouse model shown here. After using SGLT2 inhibitor, senescent cells stained by the SA-beta staining were decreased, indicating the anti-aging effects of SGLT inhibitors. And also they found that some PD-1 positive and PD-L1 positive senescent cells were decreased in the fat tissues as well. And also some few data indicating that direct effects of SGLT2 inhibitor not related to SGLT2, which means the off-target effects. You can see the in silico molecular docking analysis or isolated perfusion heart experiments indicated that SGLT2 inhibitor can bind to cardiac glucose transporters such as GLUT1 or 4 to reduce glycolysis affinity, chromatography, and SGLT2 inhibitor can directly bind to the mTOR proteins shown here. So to conclude, after using SGLT2 inhibitors in human population, regardless of diabetes, they have strong benefits on cardiovascular and renal diseases. And many structural benefits and metabolic benefits are summarized in this figure, and also many kinds of basic mechanism, molecular targets were identified from the animal studies, but still it's not fully understood, so many research should be conducted to verify the specific effects of the SGLT2 inhibitors. So most of my research were conducted by our team, including Dr. Eunlan Kim and Sora Kim, and I'd like to thank other collaborators. And finally, I'd like to briefly introduce our Diabetes and Metabolic Conference in Korea, which will be open in September 25th, this autumn. Because cardiovascular disease and metabolism are tightly linked, so I'm scientific meeting, and thank you for your listening.
Thank thank you, Dr. Lee, for the good presentation. So SH2 inhibitor possess many effects on cardiovascular disease and renal disease. So this paper is now open for discussion and comments and questions from the audience. So I have one question. So why does SH inhibitors reduce the inflammation? Could you let me know the mechanisms?
Yes, thank you, Suya, for the important question. Many animal studies and human clinical trials show the anti-inflammatory effects by the SGLT inhibitors. And because most of immune cells don't express SGLT2 proteins in their cells, so we largely think that kind of effects are indirect, and many possible mediators such as ketone body also affects, and also lowering insulin level also reduce the inflammatory signals, and also other kind of fast mimicking activity such as modulating AMP kinase or mTOR signaling may be related to the reducing inflammations.
Okay, thank you very much. So, Dr. Zhu, do you have any questions?
Okay, Dr. Lee. So thank you for your speech. You introduced so many effects for the SGLT2 on the heart disease, kidney disease, and other disease. So my question is, in fact, the short question, is there possible for the guideline to improve the position of the SGLT2 similar as the aspirin or the metformin in the future?
Yeah, that's a really insightful question. And as a clinician and endocrinologist, I hope that SGLT2 inhibitor can be positioned as a like anti-aging or like a primary disease preventive drugs. But because this kind of drug also has some side effects, like aspirin can induce like hemorrhage in our stomach. So like this SGLT2 inhibitor also has good points, bright points, but also dark points, such as side effects, such as like genital infection or dehydration. That kind of thing can happen. And also by using this drug in very weak old patients, maybe they feel really tiredness and maybe they are dehydrated and weight loss can happen. So maybe this the characteristics of this drug can be so widely used like aspirin, but maybe that indication, I hope that can be expanded to other.
Okay, thank you.
Any question, comment? Okay. Well, thank you, Dr. Lee, for good presentation. Okay, let's move on the next topics. Our next speaker is Dr. Duan from Anhui Prevention Hospital. Her title is The Role of Glucose and Lipid Metabolism Disorder in Atherosclerosis. Okay, please start, Dr. Duan.
Okay, thank you, Morimoto, and it's really my great honor to participate in this seminar, and I really appreciate the invitation from Professor Zeng and Professor Morimoto. And my topic today is about the glucose and lipid metabolism. As we know, atherosclerosis is the pathological factors in the cardiovascular disease, and there's a lot of risk factors have been found in the atherosclerosis. And the main component of the plaque is cholesterol-rich macrophage-like cells. So we know that atherosclerosis-related cardiovascular disease remains the leading cause of death in the patients with metabolic disorders, even with strict cholesterol control. So we are interested in the question, what is the role of the disorders of glucose and lipid metabolism in the atherosclerosis and what occurs in the macrophage function? And also we want to develop some new therapies. So first, we isolated the tissues from the metabolic disordered mice and crossed the genes changed. We found an interesting gene, RTN4 for short, also named NOGO. I would like to the pointer here. The name is RTN4. It's a protein on the ER of the cells. So we notice this gene and we want to identify the role of this gene in this process. So first, we would like to investigate the role of RTN4 in the metabolic disorders. As we know, the glucose can be converted into lipid in the cells. We found that high glucose can stimulate the RTN4 expression, and it can stimulate the conversion from glucose to lipid. On the other hand, the lipid can also increase the expression of RTN4. And we also observed the RTN4 was increased associated with BMI in humans. If we knock out RTN4 in the mice, we found the body weight can be decreased. Even the mice, the knockout mice take more food. So we identified the role of RTN4 in the metabolic disorders. We think RTN4 may be important in the positive feedback in this in the glucose and lipid disorders. We also found that if we knock out RTN4, the conversion from glucose to lipid can be decreased, as well as AMPK phosphorylation was enhanced. So we confirmed that the conversion from glucose to lipid may play an important role in this process. If we knock down the ChIBP, an important transcriptional factor in this process, we can not only improve the metabolic disorders in the pregnant mice, we also can improve the metabolic disorders in the offspring. So we moved to the next question. How can the RTN4 promote atherosclerosis? We use epi mice to find the protein interacted with RTN4. We also use data from the giraffes and the human and mice to confirm that RTN4 was associated with the coronary heart disease. We found that RTN4 can bind to the liver X receptor. It's a nuclear receptor controls the cholesterol efflux and inflammation, especially in the atherosclerosis. We found that they can interact with each other in the cells. And we want to know what's the function about the interaction. We use the Western blot to see the RTN4 can promote the degradation of the liver X receptor. Then we crossed the mice with the ApoE deficient mice and the RTN4 knockout mice. We found that the cholesterol levels was decreased when RTN4 was deficient, and the atherosclerosis was decreased. We also used LDL receptor deficient mice. We also found that it can inhibit atherosclerosis in LDL receptor knockout mice. As RTN4 can affect the LXR degradation, and both ApoE and LDL receptor can be regulated by liver X receptor. We use the ApoE and LDL double hydrogen to see if it can improve the inhibition of the atherosclerosis. We found that in this mice, the atherosclerosis was inhibited significantly. So we can come to the brief summary. We found that the disordered glucose and lipid metabolism can promote RTN4 expression, and RTN4 can promote the liver receptor degradation and result in the promote on the and then it can promote the atherosclerosis. Then we want to know if the anti-4 can affect other proteins. We look back on the IP mice. We found an important molecules, the pro-GCG. It is the glucagon precursor. In the pancreas, the precursor can be converted into the glucagon, but in the intestine, the pro-GCG, the precursor of the glucagon, can be cleaved into GLP-1. We confirm that RTN4 can interact with the pro-GCG in the cells, and if they interact each other, we found that RTN4 can cause the pro-GCG in the ER while they are unable to undergo the shear processing and secretion. Then we use the intestine green mice to cross with RTN4 flux mice. We generated the intestine knockout RTN4 specific mice. We found that GLP-1 in the serum was increased significantly. As we know that GLP-1 has many benefits on the vessels and the heart, and it can also decrease the inflammation in the atherosclerosis. We all know that the anti-inflammation can inhibit the atherosclerosis, but it's not very clear that if how inflammation can promote atherosclerosis. We use the model to mimic the inflammation in the atherosclerosis. We found that a receptor for the harmful cytosis, it is named the TIMD4 on the macrophage. If there are some inflammation in the cells, it can activate the ADAM17, and this enzyme can cleave the TIMD4. Then the soluble TIMD4 can be found in the circulation. When this molecule was disappeared on the membrane in the macrophage, the cytosis was impaired in these macrophages, and it can promote the atherosclerosis. We also want to know if this process also exists in the patients. We collect the serums from the CHD patients and the healthy controls. We found the soluble TIMD4 can increase in the patients, indicates the impaired cytosis. We also collect the serum from ACS patients and the CCS patients. We also found that the levels of soluble TIMD4 was increased in the ACS patients. As we know, the impaired cytosis may be earlier than the atherosclerosis, so we think this process can be a target for the earlier atherosclerosis. Then we want to develop some therapies on the atherosclerosis. We want to identify the structure of RTN4. We collaborated with the scientists in the protein structure. However, it is really difficult, so we looked through the downstreams of RTN4. As ChIBP, its structure was also don't know, so we noticed that AMPK phosphorylation. We also developed some compounds can inhibit the expression of RTN4, but it's not very directly. So we want to develop some work on the AMPK. However, we know that metformin may have little effect on the atherosclerosis, so we want to develop a new activator for the AMPK. We collaborated with the Professor Zhu. Their lab developed a new agonist of AMP called 007 for short. We use this compound to treat the mice. We found it can increase the metabolism in the mice. It can also decrease the cholesterol levels in the ApoE deficient mice and inhibit the atherosclerosis development. Then we use the LDL receptor knockout mice. We found although the cholesterol was not changed by this compound, but the triglycerides levels was significantly decreased, as well as the free fatty acids levels. Even it cannot reduce the cholesterol levels, it can also decrease the atherosclerosis lesions significantly, indicating there are some independent pathways on the cholesterol. Then we want to know if AMPK is the central role in this process. We use the AMPK knockout mice, and we found that if AMPK is knocked out, the atherosclerosis was not inhibited by this compound. In summary, we found that the disordered glucose and lipid metabolism can stimulate the RTN4 expression. One hand, the RTN4 can enhance the protein degradation of the liver X receptor and promote the atherosclerosis. On the other hand, RTN4 can also regulate the secretion of GLP-1, and we also found that the inflammation can promote atherosclerosis associated with the impaired atherosclerosis. We also developed some therapies on the atherosclerosis. We found that a new AMPK activator can inhibit the atherosclerosis in an AMPK-dependent pathway. Associated with this process, the compound can both decrease the cholesterol levels and triglycerides levels to inhibit the inflammation in the atherosclerosis. And last, I would like to make my acknowledgement. I appreciate the support from Professor Junbo Ge and my backgrounds from the NSFC, and I also appreciate all my collaborators on this research. My lab based on the Anhui province in the Anhui Principal Hospital, and here's my email. And thank you for your attention. Thanks again.
Thank you, Dr. Dian, for the good presentation. So you indicate, I have two questions. So you indicate that the RTN4B knockout mice inhibited atherosclerosis. So does overexpression of RTN5 promoted atherosclerosis?
You mean if we overexpress
Yes,
RTN4?
yes.
Yeah. Actually, we didn't do these experiments, but we observed that high cholesterol levels can increase the RTN4 expression in the atherosclerosis. We think maybe it can also promote this process. We have done some experiments in the cells. We found that overexpression of RTN4 can decrease the cholesterol efflux in the macrophage, and which may be the main process for the atherosclerosis development. Okay, thank you
Thank
for
you.
your question.
Okay, I have another question. So do you want to apply your compound IMFHO07 to clinical study?
Yes, thank you so much for your question. The 007, the compound was now in the phase 1 clinical study, and I hope it can move on to the phase 2, and we look forward for this compound. Thank you.
Thank you. So could you let us know that, Dr. Chen, could you let us know any questions?
No, I have no further question. You can close this session.
Okay, thank you very much. So time is almost done, so we would like to close this session. So we have 4 speakers and very good presentations. So I think that this session will be continued to next year. So I think I want to close this session. Thank you very much for your attention. Thank you.
Okay. Hi, Yibin and Junyi. So we will transfer the session to Yibin.
Good morning, everyone. I think that we still have a few minutes, right? Yeah.
Yes, as a plan, we will start the next session.